Before start this video, please subscribe my channel and click on bell icon. Thanks.
New Delhi, microwaving plastic food containers can release billions of small toxic plastic particles, according to a study. The researchers found that microwaving plastic baby food containers released over 2 billion nanoplastics and 4 million microplastics for every square centimeter of container in some cases. The study, published recently in the journal Environmental Science Technology, also found that 3/4 of cultured embryonic kidney cells had died after 2 days of being introduced to those same particles. It is really important to know how many micro and nanoplastics we are taking in, said Kazi Albab Hussain, the study's lead author and a doctoral student at the University of Nebraska-Lincoln, U.S. Many studies, including ours, are demonstrating that the toxicity of micro and nanoplastics is highly linked to the level of exposure, Hussain added. The team conducted experiments with 2 baby food containers made from polypropylene and a reusable pouch made of polyethylene, both plastics approved by the U.S. Food and Drug Administration. In one experiment, the researchers filled the containers with either deionized water or 3% acetic acid, the latter intended to simulate dairy products, fruits, vegetables and other relatively acidic consumables, then heated them at full power for 3 minutes in a 1,000 watt microwave. Afterward, they analyzed the liquids for evidence of micro and nanoplastics, the micro being particles at least 1/1,000th of a millimeter in diameter, the nano any particles smaller. The actual number of each particle released by the microwaving depended on multiple factors, including the plastic container and the liquid within it. However, based on a model that factored in particle release, body weight, and per capita ingestion of various food and drink, the team estimated that infants drinking products with microwaved water and toddlers consuming microwaved dairy products are taking in the greatest relative concentrations of. Experiments designed to simulate the refrigeration and room temperature storage of food or drink over a 6-month span also suggested that both could lead to the release of micro and nanoplastics, the researchers said. The team, including Svetlana Romanova from the University of Nebraska Medical Center, cultured and exposed embryonic kidney cells to the actual plastic particles released from the containers. Rather than introduce just the number of particles released by one container, the researchers instead exposed the cells to particle concentrations that infants and toddlers might accumulate over days or from multiple sources. After 2 days, just 23% of kidney cells exposed to the highest concentrations had managed to survive, a much higher mortality rate than that observed in earlier studies of micro and nanoplastic toxicity. The team suspects that kidney cells might be more susceptible to the particles than are other cell types examined in prior research. Regardless of its experimental conditions, the team found that polypropylene containers and polyethylene pouches generally release about 1,000 times more nanoplastics than microplastics. Thanks for watching. Please subscribe my channel for more videos.